Next, in this video, we are going to discuss about the antimicrobials. We know that the disease in human and animals may be caused by the varieties of microorganisms such as bacteria, virus, fungi and other pathogens. हाँ, अब ये जो माइक्रो ऑर्गेनिज्म की वजह से जो डिसीज होंगे देन इट इज़ कॉल्ड एज माइक्रोबियल डिसीज. से if we want to control this microbial disease, then we require antimicrobial drugs. Antimicrobials tend to destroy or prevent the growth of microorganisms by interfering with their metabolic pathway. Antimicrobial tends to destroy or prevent the growth of microorganisms by inhibiting with or intervening with their metabolic pathway. If the microorganism which can be treated or killed by this antimicrobial, then it's called as, if bacteria, then it's called as antibacterial, fungi then antifungal, virus then antiviral, and parasite then antiparasital drugs. All these are the category of antimicrobials. The most widely used antibiotics, disinfectant and antiseptics are also the category of antimicrobial. In this first section, we are first going to be discuss about the antibiotics. Antibiotics, as the name indicates, against the bioorganism. Against the anti means against and bio means living. Living microorganism के against means the chemicals or compound which are used to treat the infection caused by the microbes, then is called as antibiotics. This has very low impact on human and animals, but its mainly causes the activity on microorganism. Antibiotics are used as a drug to treat the infection because of their low toxicity to the humans and animals. Initially it was classified as a chemical substance. पहले जो सबसे beginning में first definition of antibiotics is like that. Antibiotics are the chemicals which is produced by the microorganism and used to kill or inhibit the growth of microorganism. हाँ, सबसे पहले ऐसा माना जाता था कि chemical substance antibiotics are the substance which is produced by the microorganism like bacteria, fungi and molds and inhibit the growth of microorganism, then it's called as antibiotics. But later on it is possible that we can synthesize or modify the chemicals which is produced by the microbes, and even in the later stage of the development we observe that there are so many number of chemicals which can be synthesized chemically but used to kill or inhibit the growth of microorganisms. So due to this, because the antibiotics can be synthesized externally or synthetically in a laboratory, so the new definition of antibiotics are come into the existence. Now we refer antibiotics as a substance produced wholly, pura completely, wholly or partially by the chemical synthesis. Antibiotics now referred as a substance produced wholly or partially by the chemical synthesis, which in low concentration inhibit the growth or destroy the microorganism by intervening with their metabolic process. So it is not necessary that it must be synthesized naturally, but it can be wholly or partially synthesized by chemical process and in a low concentration inhibit the growth of microorganism or destroy the microorganism by inhibiting with or interfering with its metabolic pathway. So the major outcome in the uses of antibiotics is started with the use of salvarsan. Salvarsan is the drug which is used in the treatment of syphilis. The scientist Paul Ehrlich played a very important role in the discovery of the salvarsan. He identified that arsenic containing compound salvarsan has a more toxic effect on the syphilis bacteria but less toxic to the human being. And based on that, he produced the drugs which is called as acifenamide. Nowadays it is called as salvarsan. In this way, the first antibiotics come into the existence, that is produced by the Paul Ehrlich, which is used to kill the microorganism. Clear? And it is used in the treatment of the disease, that is syphilis के अंदर यूज़ किया था, bacterial disease को. Now while he studied the salvarsan, he find that parallelly he was also studying the many number of azodie compounds and you can see in the diagram that its diagram is very clear. प्रोटॉन सेल यहाँ पे नाइट्रोजन नाइट्रोजन डबल बॉन्ड है और सल्वासन में आर्सेनिक आर्सेनिक डबल बॉन्ड था. द स्ट्रक्चर ऑफ सल्वासन एंड प्रोटॉन सेल इज ऑलमोस्ट सिमिलर, बट आर्सेनिक इज रिप्लेस्ड बाय द नाइट्रोजन and there are some structure modification is taken place, and based on this azodie, study of azodie, he come to that some of the azodie containing compounds show the structure similarity with the salvasan, and he tried to produce some of the agent which is structurally similar with the salvasan, and find that the proton cell is one of the nitrogen containing compound in where it can be used in the treatment of syphilis. Once proton cell is taken inside the body, it is get converted into sulfanilamide. It is get converted into sulfanilamide, SO2NH2. And this sulfanilamide is responsible for its antibacterial properties. Antimicrobial property of the proton cell is because any drugs whenever taken orally in our body in the liver, it can be metabolized, and after metabolized it converted into sulfanilamides. And this sulfanilamide is responsible for its antibacterial or antimicrobial properties. In this way, sulfur drugs is come into the existence. Arsenic is salvasan is arsenic compound, arsenic containing compound, whereas proton cell is a dinitrogen azo group containing compound as a sulfonamide groups. And this sulfonamide result into the sulfanyl after the enter in our body. Likewise, sulfapyridine is also the one of the drug which can be used in the treatment of syphilis. So in this way, antimicrobial drugs was discovered by the Paul Ehrlich, and for that he was also awarded Nobel Prize in medicine for his discovery. But this also opened the door for the antibiotics. But the main breakthrough is come when the scientist Alexander Fleming discovered the Penicillium notatum. He identified that the drugs penicillin is obtained from the fungus Penicillium notatum, and from that which can be used in the treatment of variety of disease. So in this way antibacterial therapy get started. Now the antibiotics are mainly classified into two types: bacteriostatic and bactericidal. Cidal means killing effect, which can kill the microorganism, and static means inhibit the growth of microorganism. There are two ways in which antibiotics can work. Either they may kill the microorganism or they may inhibit the growth of microorganisms. So nowadays antibiotics are mainly classified into two types: cidal, bactericidals or bacteriostatics. The drugs like penicillin, aminoglycosides, ofloxacins are the bactericidal in nature. Penicillin, aminoglycoside, ofloxacins are the bactericidal in nature, whereas the drug like ETC, erythromycin, tetracycline and chloramphenicol, ETC याद रखिएगा, ETC स्टेटिक के वर्ड में पीछे सी से स्टार्ट होता है, स्टेटिक बराबर टिक, erythromycin, tetracycline and chloramphenicol are bacteriostatic in nature, whereas cidal, penicillin, aminoglycoside and ofloxacin are bactericidal in nature. Next is important part is that the antibiotics which can able to kill or inhibit the growth of wide range of microorganisms then is called as broad spectrum antibiotics. Broad spectrum antibiotics, that is on which range of bacteria or microorganism that your drug can affect. So antibiotics that kill or inhibit the wide range of gram-positive and negative bacteria are called as broad spectrum antibiotics. Those which is effective only against the narrow range of or only a gram-positive or negative bacteria then is called as narrow spectrum antibiotics. Those which is particularly affect any particular strain of the microorganism then is called as limited strain spectrum antibiotics. The drugs like penicillin G is a narrow spectrum antibiotics. Penicillin G is a narrow spectrum antibiotics. But the structure modification is taken place in a penicillin, and the ampicillin and amoxicillin are synthesized, which are act as a broad spectrum antibiotics. Broad spectrum antibiotics. It is essential to test the allergy for the penicillin for the human beings before taking it, because some of the individuals are allergic to the penicillin derivatives. Then if we compare the structure of chloramphenicol is isolated as a 1947 and act as a broad spectrum antibiotics. It is used in the treatment of typhoid. It is rapidly absorbed from the GIT and hence can be used in the treatment of typhoid, dysentery, acute fever and certain form of urinary infections. Vancomycin and ofloxacin are other important class of broad spectrum antibiotics. The antibiotic desiderasin are used to be toxic toward a certain strand of the cancer cell also. Now you can see the structure of penicillin. Penicillin is a structure where carboxylic acid group, sulfur, nitrogen and amide all groups are present. This is a structure of penicillin. बराबर घर जैसा स्ट्रक्चर बनाने का आपको, that is a structure of penicillin. Carboxylic acid, nitrogen, sulfur and amide groups are present. Whereas in the structure of chloramphenicol, nitro group is there, phenol, chloramphenicol, chloro group is there, phenolic group is there, CHOH, and alcohol is there. So that's why it is a structure of chloramphenicol. Chloramphenicol is used in the treatment of typhoid fever. Then the next most important one is antiseptics and disinfectants. Antiseptics and disinfectants played a very important role in our day-to-day life. Normally, when we suffer from any injury, clear, we are used the antiseptics. Both of them can kill or inhibit the growth of microorganism. Both antiseptics and disinfectant can kill or inhibit the growth of microorganisms. Where antiseptics can be applied? Antiseptics can be applied to the living tissues. Living tissues such as wounds, cut, ulcer or any diseased skin surfaces. Then we need to use antiseptics. जो living animate objects है वहाँ पे. For example, furacin जो soframycin etc. are the example of antiseptics. They are not ingested orally like antibiotics. आप उसको orally ingest कर सकते नहीं हैं. Commonly used antibiotics is Detrol. What is Detrol? Detrol is nothing but the mixture of chloroxylenol and terpinol. Most time पे entrance exam के लिए भी Detrol is an antiseptic and it is a mixture of chloroxylenol and terpinol. Likewise, bioethanol are added in a soap to provide antiseptic property to it. जैसे कि आपने सुना होगा कि Dettol है, फिर Lifebuoy है, Savlon है. There are some soaps which claim that the soap has an antiseptic property or ability to kill the microorganism because they consist of the bioethanols. Likewise, the iodine is also act as a powerful antiseptic, and 2 to 3% solution of iodine in alcohol water mixture is called as tincture of iodine. Tincture of iodine. It is also used to kill the microorganism. In the gargles, they can also be used. Iodoform is also used, whereas boric acid can act as an antiseptic and used in a eye solution. जब भी आप कंजक्टिवाइटिस होती है, बराबर कंजक्टिवाइटिस होते हैं तो बोरिक एसिड सॉल्यूशन, डाइल्यूट सॉल्यूशन ऑफ बोरिक एसिड एक्ट एज ए वीक एंटीसेप्टिक फॉर द आई साइड पे इन्फेक्शन होते हैं तब उसका यूज़ किया जाता है. बायोथायोनोल 2 बेंजीन रिंग है, 2 फिनोलिक डेरिवेटिव्स कनेक्टेड बाय सल्फर इज कॉल्ड एज बायोथायोनोल. टर्पिनॉल साइक्लोहेक्सिन डेरिवेटिव्स है. क्लोरोजाइलोनॉल, जाइलेनॉल यानी जाइलोन यानी मिथाइल ग्रुप होने चाहिए, फिनॉल एंड क्लोरीन ग्रुप, क्लोरोजाइलेनॉल. द स्ट्रक्चर याद रखने हैं, एग्जाम में स्ट्रक्चर पूछे जाते हैं, एंट्रेंस एग्जाम में स्पेसिफिकली. विच ऑफ द फॉलोइंग इज द स्ट्रक्चर ऑफ क्लोरोजाइलेनॉल. बोर्ड में नहीं पूछे जाते हैं, एंट्रेंस एग्जाम में विच ऑफ द फॉलोइंग इज द स्ट्रक्चर ऑफ टर्पिनॉल. बायोथायोनॉल 2 बेंजीन रिंग, 2 रिंग होंगी, ध्यान रखना है. सो दिस इज अबाउट द एंटीसेप्टिक्स. तो सर डिफरेंस क्या है एंटीसेप्टिक एंड डिसइंफेक्टेंट में? एंटीसेप्टिक लिविंग टिश्यू पे अप्लाई होंगे, वुंड पे, अल्सर पे, इंजरी है कहीं पे तो उस पर बराबर. बट डिसइंफेक्टेंट मेनली अप्लाई ऑन इनएनिमेट ऑब्जेक्ट्स, नॉन लिविंग ऑब्जेक्ट्स पे अप्लाई करना है तो हम डिसइंफेक्टेंट का यूज़ करेंगे, सच एज फ्लोस, ड्रेनेज सिस्टम, इंस्ट्रूमेंट, सर्जिकल इंस्ट्रूमेंट, उन सबको डिसइंफेक्ट करना है तो डिसइंफेक्टेंट यूज़ किए जाते हैं. सम ऑफ द सब्सटेंसेस इन द लो concentration act as both disinfectants as well as antiseptic. For example, 0.2% solution of phenol act as a disinfectant, antiseptic, whereas 1% is act as a disinfectant. Similar way, chlorine in the concentration of 0.2 to 0.4 ppm in aqueous solution and sulfur dioxide in a very low concentration act as a disinfectants. This is about the antiseptics and disinfectant. Hopefully you guys enjoy the video. Please do comment on the comment section.